TB or not TB? So you always have faced a question when you're working with bacteria, what do you feed them? So TB, terrific broth, and LB, lysogeny broth, are two of the common bacterial media or food that we use to feed bacteria. There's also other types of media that we use, and we can use media to do things like selection and screening and various things. So today I want to tell you about all of the variety of great things and cuisine to choose from when you're working with bacteria, getting them to make DNA and protein and all that great stuff.
Biochemists get bacteria to do a lot of work for us. We can get them to copy DNA if we stick the genetic instructions for making a protein or something into a piece of circular DNA called a plasmid, and we stick that in bacteria. The bacteria will make a ton of copies of it that we can then purify out. So we can get them to make lots of copies of DNA, and we can even get them to make protein from that DNA that we put in there. And we can even do this with this inducible system where we can get protein made on demand, as I talked about in my post yesterday. But they don't do this work for free. We have to feed them. So this is our media making room where our amazing media maker, Adriana, makes the media for the bacteria, and the media is their food. So there are different types of media, and they're good for different purposes. So for example, here's SOB, TB, and LB. We'll get more into what all of these are. The main one is LB, lysogeny broth. So this is the main one, and we'll talk about it in detail first. We can have their media just in like flasks or in bottles, or we can have their media embedded in an agar gel in a petri dish. So one of these like circular plastic dishes, we can make this gel filled with bacteria food. And so this gel is made out of this sugar agar. So agar is a mix of agarose, which is the sugar that we use when we're we're separating DNA fragments by size using electrophoresis, as well as agaropectin, which is shorter and it's modified kind of randomly and charged. And this charge and variability and composition makes it so that you wouldn't want to use it to separate DNA, but it's really good for making bacterial pancakes, these agar media plates. So a fun fact about these agar plates. So apparently the first gelling agent that was tried was by Robert Koch in 1881. He tried gelatin, but the bacteria ate it, and it also melted too easily. And so the wife of one of his associates, Angelina Fanny Elshemus, suggested that he try agar instead, and it worked well. The bacteria didn't eat it, it stayed solid, and it's going to kill the bacteria. So from thenceforth, we have used agar plates, and we have Angelina to thank. So thank you, Angelina. If we add antibiotics, we can select for bacteria that we want. So when we're doing biochemistry and not just like trying to culture whatever bacteria is hanging around, we want to make sure that only bacteria that actually have the DNA that we put in there are going to survive. And so we can do this by selecting using this antibiotic resistance gene on our plasmid. So we put that in as well as the gene or the whatever information we want in the plasmid. And this way, when we spike their food with the antibiotic, we're only going to allow the ones that survive that have the plasmid. The ones that survive are going to grow on top of themselves in these colonies, these like loopy dots, and then we can isolate stuff from there if we want. But basically, we only want to feed the ones that we want to survive. We also see this media a lot in flasks, and we can grow bacteria in suspension, so like in this liquid as well. And this liquid is what's mixed with the agar when you make the gel. So let's look at what's in this liquid. So first, let's clear up the name. So it was invented by Giuseppe Bertani in 1951, and he was working in the lab of Luria. So sometimes you'll see it called like Luria broth or Luria Bertani broth, sometimes Miller and Bertani. So Miller is this guy who just basically wrote this textbook and stuff with all these recipes. And so he put in the recipe for the LB and then kind of got credit. But really it's Bertani that is the person that we really have to thank for this. And he called it lysogeny broth. So lysogeny he called it that because he was studying lysogeny, which is where it's just like phase in some viruses life cycle when they infect bacteria, and they kind of just like hang low for a while. And then they hang low and they replicate and stuff. And then the bacteria later, when they're going into a lytic phase where they're actually going to like break open the bacteria, and that sort of thing. But when he was working in this lysogeny phase, so he was studying this, it's lysogeny broth. This is what he was growing it in. So what is in it? So basically it has things that the bacteria need to survive and thrive. It has tryptone. It's a type of peptone. So peptones are a mix of protein pieces. So proteins are like polypeptides. You cut them up and we call these like peptones. And so peptones are a mix of protein pieces that come from chopping up proteins. So tryptone is when the chopped up protein is casein, and the choppers are pancreatic enzymes. And it's a good source of amino acids for making new proteins that we want the bacteria to make for us, plus the ones that they need themselves. We have yeast extract. So this is basically whatever was in the yeast. So we just break open the yeast, take what's in there. So I mean, if it's good enough for the yeast, right? It's good enough for the bacteria, probably. So this is where you get your source of vitamins, trace elements, etc. Salt. So this has sodium chloride, and here you'll see some differences in various formulas for LB. And so Miller and Bertani, they have like a ton of salt in there. It's like 10 grams per liter. Lennox is like half of that, 5 grams per liter, and Luria is only like 0.5 grams per liter. So these differ in the amount of salt, and why this might matter is that if you're working with a salt-sensitive antibiotic, you might want to use one of the lower salt formulas. As well as for other various purposes, there are reasons why you wouldn't want a high amount or a low amount of salt. So Bertani, his formula also includes glucose. And so like the original formula has glucose, but nowadays the formulas typically leave out the glucose, although as we'll see, there are media where they have glucose in them. But Bertani added glucose, and he added it after the autoclave step. We'll talk more about the autoclave later. But because it would undergo like caramelization and the Maillard reaction and stuff, and react with the proteins and stuff, and this was inhibiting some of their bacteria stuff. But anyway, most of these formulas leave out the glucose these days. So you've got the protein pieces, you've got the like vitamins, minerals, that sort of thing. You've got salt to keep with the osmotic balance, help with the transport, etc. And then in some cases you have the glucose. That's like the basic formula, the basic media that is the groundstone of a lot of what we do. So we often have like, just when we're just doing normal types of work in molecular biology, biochemistry, whatever, that's kind of like the go-to. But sometimes you want to have different media for different purposes. And so if we think about a couple main purposes of using bacteria in biochemistry are to make proteins and to make DNA. So when you want to make protein, you're going to want to have like a rich media, lots of peptones, lots of that sort of thing. Where you have those protein pieces that you can be using as a source of amino acids and all these nutrients and that sort of thing. And so the one that I use a lot is TB or terrific broth. And so this has more yeast extract. So if it has a green arrow, that means that it has more of this thing than the LB does. And if it has a star, that means it's something that the LB doesn't have. So in your TB, you have additional yeast and additional tryptone, and you also have these these potassium phosphate buffers. So this is going to help stabilize the pH to prevent cell death. So you have a richer media. It's going to allow for more protein production, higher yield in a shorter amount of time. Sometimes the cells can grow to a higher density, and you have this prevention of cell death. So this can be really good for making proteins. So with TB, you see that we have, so this is TB, and this you'll see is 10x TB salts. So you actually add these salts in after you autoclave. So autoclave is where we do this like heat sterilization. And when we do this, it gets really hot and really high pressure, which is going to sterilize things, but it can also cause chemicals to react with one another in certain cases, and chemicals to break down in certain cases, and that sort of thing. So if you have these TB salts in there, if you have this like potassium phosphate in there when you're doing the autoclaving, if you have it in the mixture of this, so basically you want to autoclave these separately and then mix them. The potassium phosphate can actually precipitate out some things in the broth, and this can cause problems because you're precipitating things out. Another type of media that we have is this 2 times YT. So that's 2 times the yeast and more tryptone as well. So this has yeast and it has tryptone. And so this is good for long growth times. So it allows for extensive growth because you have more of this stuff to allow you to grow longer. So these can be really good for some protein cases. I'm not saying that they can only be used for protein cases, but you want different things when you're want the bacteria to be focused on making DNA. So one that we use a lot, so one thing that you want when you're making DNA is you'll want magnesium sulfate. So magnesium sulfate is really important for DNA polymerase. It is going to help, magnesium is going to help kind of in the active site of the DNA polymerase, kind of help bring together all like the negatively charged nucleotides that are coming in with their phosphate groups, and kind of stabilize things, stabilize the intermediates and help with things. So you need magnesium. And so you can provide extra magnesium in SOB and SOC compared to LB. They also have extra tryptone, so you have more of those nutrients and some other salts. And so this is good. So these two are good for DNA copying. What's different? So SOB stands for super optimal broth. I know it's a really super name, right? SOC stands for super optimal broth with catabolite repression. What is different in SOC compared to SOB is that in SOC you have glucose. When you have glucose, this is going to make it good for post-transformation recovery. So transformations where we basically get bacteria to take in a piece of DNA like a plasmid. We often do this with heat shock. So we take these these chemically competent cells, so they're really, really weakened, and then we dunk them in warm water. They get pores open up and they let the DNA in. But then we need to kind of let them recover afterwards. And because it's a big deal, kind of, they're already weak and now you're like heating them up and forcing DNA in them. So you want to let them recover. And sometimes if they have trouble recovering, and SOC can help them with their recovery because it has this glucose. So kind of like how a runner might take some glucose, like one of those gel sugar packs or whatever, to help them recover after a run. We can help these bacteria recover by adding glucose. The name, so this catabolite repression. So if catabolite is a breakdown product, so catabolism is the breakdown side of metabolism. So when you're breaking down things, you're generating catabolites. So bacteria have preferences about what they prefer to use for an energy source, and they really like to use glucose. And so this is, for example, why they have that lac operon system with the lac repressor, where they only make a lactose breaker downer if the beta galactosidase, if they don't have glucose that they can use easily, and if there's lactose around. So, but they prefer to use glucose directly. But when there's not enough glucose, they'll start breaking down other things like lactose, or maybe even various other things and forming various catabolites. You want to give them extra glucose so that you prevent them from breaking down all the other stuff and instead allow them to have plenty of energy that they need to focus on making your DNA and healing. So you note that those are all examples of what we call like undefined media. So they have batch to batch variability. So for example, like in our tryptone, that's going to have variability because basically you're chopping up the protein casein. So this milk, this protein that's present in milk, and you chop it up with pancreatic enzymes. And so you can imagine that you're going to get like a different mix every time, slightly different mix. What about the yeast extract? Well, from yeast to yeast, you're going to have variability. And so this extract is going to have variability. So the good thing about working with these types of things is that, like, when you have the yeast is going to have everything the yeast needs. And so it's going to have things, even if there's things in there that you don't know that the bacteria would want, you have it, and you have like ratios that are sustainable for life typically and that sort of thing. But it's not good in the sense that there could be batch to batch variability, as well as the fact that you can't test the importance of various things if they're already in the media. So this is where people turn to defined media. So defined media, they have constant components. So an example is M9 minimal media. So M9 media contains essential salts and nitrogen. So it has sodium phosphate, potassium phosphate, ammonium chloride, and sodium chloride. So it's offering a source of salt as well as nitrogen in the ammonium chloride. If you're like me, you're wondering where the heck the name M9 minimal media comes from. Where does the M9 come from? So there's 4 components. That's not where it comes from. Where does this M9 come from? And so I am going to disappoint you in that I still don't know. I'm guessing that it is the 9th thing that someone tried. I got really excited because I found this citation to Miller in this 1972 textbook. And I realized that I couldn't find it online, but I found that our library had copies of it, and there was even a copy upstairs. And so I rushed upstairs this morning and I found the book and I got all excited. And I looked, and in the appendix, they have this recipe for this M9 medium, but there was no index or anything, so I couldn't find the first place where was that referenced in the text itself. So I started flipping through this hundreds of page book to see the first time I saw it. And the first time I saw it, this isn't necessarily the first time they mention it, but the first time I saw it when I was flipping through, they just mention it and they don't say why. So that was a disappointment, although I did get a little smile when I saw this line, never mouth pipette radioactive solutions, which seems very logical. But anyway, so that's the minimal media. That's one example of a minimal media. So M9, that's just like minimum salts and stuff, but that doesn't mean that there's not other things that these bacteria need. And so you often add other ingredients, and you can add ingredients to test their importance or to test the ability of bacteria to survive with or without them. And so you often add things, and then this can help you test for the importance of these things or the ability of the bacteria to grow without them. So bacteria have different growth requirements, and we can use different terms to describe whether or not they need something. So imagine that you have this bacteria and it needs coffee to survive. So coffee is essential. But one of them is autotrophic for coffee, so it can make its own coffee. And one of them is oxotrophic for coffee, so it can't make its own coffee. It needs to be brought coffee. So if you grow them in media that already has coffee in them, they're both going to grow. But if you grow them in media that doesn't have coffee, now only the one that is autotrophic that can make its own can grow, whereas the one that doesn't is the one that's oxotrophic cannot grow. And this is a silly example, but it's the basis of this idea is the basis of very cool tests that we can do, such as the Ames Test, which basically tests the ability of something to act as a mutagen, so something that can cause causes changes in DNA, causes mutations by not necessarily causes cancer. This is all just happening in addition bacteria. So you take bacteria that have this original mutation in this gene that allows them to make the amino acid or the protein letter histidine. So these His minus bacteria, they aren't able to make histidine, but they need histidine to survive. So if you grow them on food that already has histidine, then they can grow fine. But if you grow them on food that doesn't have histidine, then they can't grow. And that is, they can't grow unless they acquire a mutation that allows them to make it themselves. So they go from being oxotrophic to being autotrophic, and then they're able to survive. So this is like a reversion mutation that kind of fixes that original mutation or gives them some other way to make it. And so mutagens are going to make this reversion more likely. So if you have something that's mutagenic and you add it, then you're more likely to get more of the reverted mutations. So with bacteria, as I said before, we often use antibiotic-based selection. So just a couple terminology notes. Selection is basically where we are only allowing certain bacteria to grow if they have something. So in this case, we are only allowing the bacteria to grow if they had a mutation that allowed them to, a reversion mutation that allows them to make histidine. So that's a form of selection. There's also a thing called screening. And so an example of this is the blue-white screening that I talked about the other day. In this case, you're just showing differences between the bacteria. So in this case, the bacteria look one color if they don't have the plasmid, and a different color if they do have the plasmid. So in this case, you put your gene, you insert your gene in a way that disrupts part of this beta-galactosidase gene that's needed to make this product, to convert a colorless product to a blue color. So if you insert your gene in a way that disrupts that, now you can't make that blue color and you have a white colony. But you'll still see the ones that made the blue colonies, the ones that don't have your insert, you'll still see those, they'll just show blue. So you see a difference between them. So you can screen them, but you're not actually selecting, so allowing them to selectively grow. No matter what media you're using, you're typically going to autoclave it to sterilize it. Note that when you put things into autoclave, they can change color, and you'll see some color to color variation between batches. It has to do with how long they were in the autoclave and that sort of thing, as well as what's inside them. So if you have like glucose in your media, that can cause like caramelization reactions where the glucose is reacting with itself, or you can have like Maillard sort of reactions, like browning reactions with meat type of thing, where you have the sugar reacting with protein. And so you can get these color changes to happen. And it's also, depends on what's in the media and how long it autoclaves for. But don't be worried if you see like, oh my God, this one's a little darker than this one. That's all that it is. So yeah, like in undergrad, I was making all my own media, and now we have this media maker here, and Adriana is amazing. So thank you, Adriana, and thank you to all the media makers out there. You are greatly appreciated. So they make all this great media, and then they stock our cabinets for us, and we can just use it when we want. We're really fortunate with this, and I am very, very grateful. And also, it's probably an acquired thing, but I really love the smell of bacterial media. And yeah, but I wouldn't want to eat it, but bacteria want to eat it. So that's good because we can feed it to them and they'll be happy and do things for us. So happy biochemistry with bacteria.